PowerPoint?
Yeah, that sounds good.
They can't multiply unless they invade a specific host cell, and they must instruct the genetic and metabolic machinery of host cell to make and release new viruses. They're inactive macromolecules outside of the host cell, and they have the basic structure of a protein shell, which is a capsid surrounding a nucleic acid core. They're ubiquitous in nature and have had major impact on development of biological life. They're ultramicroscopic, can have either DNA or RNA, never both. They carry molecules on the surface that can only attach to the specific host cell. They multiply by taking control of host cells' genetic material and regulating the synthesis and assembly of new viruses. They lack enzymes for most metabolic processes and the machinery for synthesizing proteins, which is ribosomes.
All right, so we can do the concept check if y'all want. The first concept check is that viruses are alive. It's a true or false. And we agree that it's false, right? Okay.
Yeah.
Yes.
And it says that we can discuss the predominant view among scientists as to the nature of viruses. So I know that they're specific, host specific. I think that's what it's referred to.
Yeah, I think they're like obligate intracellular.
Right. So they can infect like every different group, but each virus is specific to one type of group and it can only infect that type of group.
They also thought, like, before they knew about viruses, they thought that it was just any infectious agent. They didn't know the difference between like virus and bacteria until filterable viruses.
Mm-hmm. That's good. Um, let's see. It says what percentage of the bacterial chromosome is composed of viral genes, but I don't know the answer to that.
I don't know this either. Yeah, I'm not quite sure.
I
It's
think it was like
composed of
8%, maybe.
8%.
I think, don't quote me on it, but
I can look it up.
Well, that would be perfect. Could you please?
Yeah. It says 10 to 20%.
okay.
Yeah.
And then 5 unique
This makes
properties
sense.
of viruses. Well, that they're not cells and
Yeah.
that they're not
Yeah.
alive, but they have some characteristics of living things.
right. And they cannot multiply without a host.
Mm-hmm.
Protein shell and nucleic acid core.
They're the smallest.
Right.
And they also only have DNA or RNA. They don't have both, which I think is a really big thing for them.
Yeah.
I think that we've hit 5. So we can go on
Yep.
to the general structure of viruses. So we can skip the learning outcomes and go to viral size. So they're 20 to 200 nanometers. And for reference, bacteria are 2000 nanometers and microscopic. So viruses are like very, very tiny. And that explains why they pass through filters that don't let bacteria to pass through them. And we can't see them without electron microscopes. And they're not made of cells. So their components include a capsid, which is the shell that we talked about made of protein subunits, and it surrounds the nucleic acid. The nucleocapsid is the combination of the capsid and the nucleic acid together. Next, we have the envelope, and not all viruses have an envelope, only some. And then we have spikes, which are found on both the naked and the enveloped viruses, and they project from the nucleocapsid or the envelope, and they allow viruses to attach to the host cells. And a naked virus consists of only the nucleocapsid, not the envelope. And a viral particle or a virion is a fully formed virus able to establish infection in a host. So we went over the spike, the capsid, the nucleic acid, and those basic components. So the protective outer shell is the viral capsid, and it includes capsomeres, the helical capsid. I'm gonna butcher this pronunciation, but the icosahedral capsid, and then complex capsids. And the viral envelope is made of the cellular membrane of the host, and regular membrane proteins are replaced with viral proteins. And then the spikes are made up of glycoproteins, and they're essential for that attachment to the host.
Okay, and then we have the nucleic acids at the core of a virus, the genome, which is the DNA carried by a cell, which is a blueprint for everything. And then the viruses can either contain DNA and RNA, but not both, like we talked about in the concepts. The number of viral genes is so small compared to that of any cell, and it only processes or possesses only the genes necessary to invade host cells and redirect their activity. And then other substances that they do not have enzymes for the viruses, and some viruses have their own enzymes which carry them inside for specific operations within the host cell, which means that the cell cannot work for some viruses without those enzymes. And then reverse transcriptase synthesis DNA from RNA, and they're only found in viruses whose genome is from RNA form. And then substances taken from the host cell and packed in the viruses are like the arenaviruses packed along the host ribosomes, and the retroviruses that borrow the host's tRNA molecules. And then this is the concept check. Do y'all want to do this one?
I'm just gonna show what I have on the concept map so far. If that's okay.
Yeah, that works.
Yeah.
Definitely.
Okay. I don't know how y'all can see me, but can the screen recording see me? Oh, okay. So I have virus and prions so far, and I just have a lot of the components of viruses, and then kind of what each component consists of as you get further down the map.
I think it looks really good.
Yeah, it looks good.
Thank you.
And we can do the concept check too.
Oh, yeah, let's do it.
Okay, true or false is viruses can be seen with a light microscope.
True, I think.
True, I
That's
believe.
true. I think that's
Yeah.
false.
Only with the electron microscope.
Right. I think it's only with the electron. I think the
Oh, okay,
is
okay,
for
okay.
bacteria and for things that are larger than viruses.
Wait, so what can you see with the light?
Bacteria.
Okay.
And because a virus is like the smallest
Right.
particle that we're studying. So anything that's bigger than anything else can be seen with the light microscope, except
Yeah.
for viruses.
Right.
So
Yeah.
viruses need a scanning electron and then a transmission electron. Those are the two microscopes they could use.
Yeah.
Yep. I just looked it up. It said the transmission electron microscope only.
Oh, okay.
Yeah.
Interesting.
So this one would be false then, right?
Yep.
Yeah.
Oh, okay.
Okay, then viral capsids are constructed from blank subunits, protein, lipids, or carbohydrates.
Protein.
Proteins.
For sure.
Okay, and then we're describing structures of round, rod-shaped, and complex viruses.
Describe the structures.
They kind of describe themselves. A
Right.
rod-shaped structure is
I think
Which one's the 3D look though?
the rod one.
icos.
Okay.
Yeah.
Is the icosahedral is the 3D
Yeah.
one. It's the 20-side figure with 12 evenly spaced corners to form
then
the
the
It forms a
complex.
like round ball.
Mm-hmm. And the rod-shaped ones form a continuous helix around the nucleic acid, and the complex ones are found in bacteriophage. Sorry, I just got
Okay, cool.
And I think the complex ones have like both the round structure, like the icosahedral, and the helical structure.
Yeah, I think that is true. So they look very complex and different. Then list 3 unique forms of a viral nucleic acid.
Wait, what?
Would those be the
I'm confused.
the forms in number 3?
Weren't those, yeah, the 3
It says viral nucleic acids can be DNA or RNA, double-stranded or single-stranded. Yeah.
Okay, that makes sense though. I didn't know really what they were asking. I think we all have a good summary of what viruses are about,
Yes.
Yeah,
Yep.
I think we're good for this concept check.
right? Awesome. Should we stop recording?
Should at 10 minutes?
Mhm.
Yeah, it's hit 10.
Okay, then yeah, we can start recording.